I will now focus on the InChildHealth consortium and how the SOPs we developed, and I will go through them. And as she mentioned, and we can go to the next slide, please. Different groups, we measure different pollutants, we use different protocols, sampling media, time frames, and quality assurance procedures. So the first, or the three main aims that we had on the development of the SOPs, the first one was to actually make indoor air quality measurements comparable across different European cities and building types, so we can ensure that results can be combined with confidence. For example, if we align the sampling intervals between classrooms and across the countries, then we remove actually one of the major barriers that we have for data comparability. The second one is to generate multi-parameter datasets that will allow us to assess in-depth pollutant characterization and also to have a robust exposure assessment. And third, to help standardize indoor air quality research in areas where internationally accepted protocols are still lacking. Next slide, please. So during the InChildHealth campaigns, we measured an extensive set of parameters that captured both the physical, the chemical, and microbiological aspects of the indoor environment. For the physical parameters, we monitored particulate matter, namely PM1, 2.5, 4, and 10. We also measured ultrafine particles, black carbon, along with temperature and humidity, which influence not only the temperature and humidity, they influence not only the comfort, but also the particle behavior, the chemicals behavior, and the microbial behavior. So there are very important parameters also to keep track of. For the chemical measurements, we included major gases like carbon monoxide and carbon dioxide, plus volatile organic compounds. We also did active air sampling and passive dust collection, where we used them to assess semi-volatile organic compounds like plasticizers, flame retardants, biocides, and fluorotelomere alcohols, which is a class of PFAS, and also polycyclic aromatic hydrocarbons. For microbiological analysis, we targeted both bacteria and fungi through culture and DNA-based methods, and we looked more specifically into Aspergillus and Mucorales species, azole resistance, Staphylococcus aureus, including the multi-resistant Staphylococcus aureus, endotoxins, and ATP for hygiene assessment. Next slide, please. So to manage all these measurements consistently, the SOPs form a continuous pipeline. So every step from field sampling to transport to extraction and analysis includes quality control checks, as blanks, for example, duplicates, spikes, field, lab, and data logs, and also metadata collection. This framework was designed to guarantee clean, traceable datasets that can be merged across the sites for exposure modeling and international comparison and evaluation. However, and Louise will mention this in her presentation, a specific SOP for data integration that considers the calibration steps for the sensors measurements, which aims to ensure data comparability across the involved partners, is currently missing, but it is foreseen to be written. Next slide, please. Thank you. So each SOP was designed to balance scientific rigor with field practicality, so they could be easily implemented across the 7 European countries. Initially, a small expert team drafted the first version, that was then reviewed independently by others with topic expertise. Then, a central coordination group harmonized the comments into a final and uniform format for all SOPs. This 3-step initial process ensured that each protocol is scientifically sound, feasible in real settings, and it is consistent with the rest. The most important part of the SOPs is that they are considered living documents. So updates occur whenever we have a field experience or quality assurance checks that reveal parts of the SOPs that need improvement, and all revisions are recorded in a traceable version history. So each SOP was also tested during the InChildHealth campaigns, and protocol adjustments were also made. And this took us more or less 1 year and a half in the process. So next slide, please. So these next 3 slides, including this one, we will go through the complete overview of the 16 SOPs that we have in our InChildHealth package. The first 2 SOPs are in the general category, and they provide a framework with the overall quality assurance and quality control principles. And the second one, it guides the integrated exposure assessment. On the physical parameters, we have 4 more SOPs, which include the measurements for the fine dust, black carbon, that actually has 2 different instruments being used, and ultrafine particles. In the next slide, we have the 3 SOPs that relate to the chemical parameters, and they detail the active air sampling and extraction of the cartridges, and also the passive dust analysis for the semi-volatile organic compounds. And finally, on the 3rd slide, we have the 6 remaining SOPs that relate to the microbiological contamination assessment, which includes sampling, extraction, and sample analysis in microbiological labs via culture-based and molecular methods, and they target different parameters. And also we have the last one is for the adenosine triphosphate testing for cleaning effectiveness. So this is the overview of how the SOPs look, the structure. Okay. And you can see that on the cover, it's very small, but there is the title of the SOP, and below we have the number of the SOP, the version number of the SOP, and also the date in which that version came into place. And then on the second page, we have 2 tables. And the tables contain information of all the authors and reviewers that were involved in the process of this SOP. And also you have the version history log. And then, of course, we start with the SOP, the scope, the methodology, and everything that we have in the SOP. Then on the next slide, please. I will go a little bit more just to show a bit more detail, and I will use as an example the microbiological parameters SOPs. I will only focus on the first one and the second one. So the first one, the 10th, number 10, is for the sampling methods. And then we have the 11th, that is for the extraction methods. And then we have 4 dedicated SOPs for the sample analysis. So if we go for the first SOP, which is the sampling SOP, I will give you can find several methodologies that are used during the campaign, from active sampling to passive sampling processes, but I will focus on the use of electrostatic dust clots, which was the one that we used commonly among all involved partners in the InChildHealth. And we have 2 types of EDCs. We have type A, and you can see them both in the picture, and we have the type B. So although they look the same, they are different. They undergo different pretreatment before the analysis. The type A is sterilized under UV light for 20 minutes, and the type B is under dry heat for 6 hours and 160 degrees. And this is because they are then later used for the assessment of different parameters. Then they undergo sampling, which is the same, more or less the same strategy. They are exposed in the classroom or in the homes for 1 month. But then again, they have a different flow on the application part. The type A is stored in cold conditions till it is extracted and processed, and this has to be done within 4 days. And after that, samples are again stored and then shipped to the partners that will process the samples. Whereas the type B is, after sampling, is just stored at room temperature in a paper envelope that was also previously sterilized in the same conditions, and then the samples are shipped at room temperature to the partner. So, and you can find the first part of the process in the SOP number 10, which is the one for the sampling, and then again the second part on the SOP 11. But one common part on both of them is actually the storage conditions, and again we have an overview of which type of EDC we are dealing with. Then for the next slide, please. We have the strategy for the sampling, so the SOP number 10. We have also the scheme on where to place the EDCs in the rooms, what rules to follow, and then we also have a table for data collection, and it immediately refers, and it's highlighted in yellow, which SOPs you need to go to search for further information on the extraction, for example. Next slide, please. So For the SOP number 11, we have the extraction methods that were used. And for the EDCs specifically, you can see that they, of course, they undergo different extraction and processing methods, but all of them are compiled and you can find all the information in the SOP. So all of our SOPs follow more or less this structure, of course, depending on the type of procedure that we are describing. But we try to make them as more inclusive and specific as possible for both experts, but also non-experts in the field. So the next slide, please. So considering the future expansion and the looking ahead, one update that should be, of course, the development of SOPs for data calibration and integration, ensuring that the sensor and the monitoring data can actually be harmonized. And my colleague Luis will explore this more. We also see potential to expand the monitoring into other child-relevant environments, such as healthcare facilities, sports hall, or public transport, so we can have a broader range of everyday exposure, so a bigger overview that goes beyond schools and homes. There is also potential to adapt the procedures for diverse contexts, which including, for example, remote or extreme environments like the Arctic communities or urban pollution hotspots. And building on our experience across the 7 European cities in the InChildHealth project, this could also enhance the geographic and environmental coverage that we have. On the chemical side, we analyzed fluorothalonil alcohols, as I referred, which are PFAS precursors, but future efforts could also include a wider range of PFAS and other emerging pollutants, which would help identify new risks earlier, ideally, and better understand mixtures and behaviors impacts on health. And finally, but not less important, is the citizen science approaches that could actually be very helpful to support more reliable monitoring in regions which are difficult to access on a regular basis, right? So it would also, at the same time, be offering STEM education and learning opportunities, building local capacity, and increasing awareness of indoor air quality. Next slide, please. To make the results comparable across the involved European countries, the InChildHealth follows the harmonized approaches that we mentioned, but we have more. And these SOPs allow with standardization concepts, but remain flexible enough to allow our some regional and national adaptation. They also support consistent sampling, quality assurance, and data collection, which enables and supports the data integration. Through shared training and common databases, we also aim to contribute to an infrastructure for large-scale exposure assessments and the identification of vulnerable population groups. This consistency directly addresses the current challenge of harmonizing indoor monitoring practices across Europe. Next one, please. And currently, Europe has several initiatives that target or at least include indoor air quality. We have the national standards and the Horizon Europe initiatives like the Ideal Cluster. And there is also the development of the ISO and the same technical standards. Also, there are the recent revisions of the European Air Quality Directive that is related with the Green Deal. And these open, of course, windows that could help to integrate indoor air monitoring and child-focused exposure indicators also. So to this opportunity, we think that the harmonized SOPs should or can be translated into practice through close collaboration between researchers, public health agencies, and policymakers. And within the Ideal Cluster, the InChildHealth SOPs and other project outputs have already been used to support the co-development of policy briefs and the evolving indoor air quality guidelines that target schools, homes, and vulnerable groups. Going forward, the collaboration with large partnerships such as PARC, which is the Partnership for the Assessment of Risks from Chemicals, will also allow these SOPs and datasets to feed into European risk assessment frameworks and support prioritization of chemicals like the PFAS or other semi-volatile organic compounds, for example, and also hopefully inform future European-wide indoor air quality standards and building regulations. So all the SOPs, next slide please, will be available via Zenodo, and of course they are free to use by all interested parties.